Hello, welcome to the Biomedical Informatics faculty presentation presented by Center for Health Informatics and Bioinformatics. I have the pleasure to introduce today Dr. Jinhua Wang. And Jinhua's been with us since the founding of CHIBI. He did his undergrad work at Beijing University and got his PhD from the Chinese Academy of Sciences Biophysics Division in the area of genomics. He did a postdoc at Cold Spring Harbor Lab under Dr. Michael Zahn, and then he worked at St. Jude's Children's Hospital. At NYU, he is an assistant professor in the Center for Health Informatics and Bioinformatics and associate director of the Cancer Center Bioinformatics Center. He is an expert in CHIBI in the areas of cancer variant detection and genomic data integration, which is almost exactly the title of his talk today.
Thank you for the introduction. Okay, today I'm going to talk about cancer genome variant profiling and integrative analysis. So we know that human genome, the normal human genome and a cancer human genome, there are many, lots of variations. So these variations gives us diversity of individuals, and also is associated with disease susceptibility. And this is a common polymorphism variants. And for rare non-polymorphism variants, those are always usually associated with for cancer with a cancer genesis and also for cancer progression. So this is a nice picture. Actually, we can see there are many types of genome variations. So these are inversion. And here, for this one, you see the translocation of chromosome 3 and 7. And also, there's another translocation events. And also, you see copy number loss and duplications, and some inversions. And this is another copy gain duplication. This picture, you have actually 10 copies of this gene, 12 copies of another gene. It covers the region of about 400 kilobase pair to actually this one is a little bit longer, 700 kilobase pair. So we can see that it's kind of a wide range. You can have single mutations to the whole chromosome abnormalities, and for copy number analysis, basically there's focus on this, from 1 kb to maybe 3 to 4 megabase long segments gain or loss. And also from sequencing, we can actually discover, single sites mutations, and also some dinucleotide insertion deletions, some short region inversions, and also some sort of variant number of tandem repeats. So actually, there are many different platforms. Like we have, array platforms to detect copy number variation. We have sequencing technologies to detect the mutations. With improvements of technologies and the growth of, these technologies, there will be more of these discoveries. So copy number variation will cause the disruption of genes in different ways. So there are actually 4 major categories that copy number events will disrupt the gene function. So deletion of one copy of a gene will actually, for dosage sensitive genes, will give a low expression of that gene. Also, deletion of a gene sometimes will unmasking recessive mutations. So when you do your sequence study, you get mutations. You probably want to also look at the copy number status to see whether there is something like this situation there. There's other type of disruption of gene functions. Like this inversion happened for this copy of this gene and actually will disrupt this gene expression. There's a deletion, a partial deletion or translocation of a gene. This part of this copy of the gene will be translocated to another part. Then maybe this promoter will be fused to a different gene. So you will get disrupted, gene regulation. And also there is a situation that genes will actually, the copy number, the deletion will occur in the promoter region, and this regulatory elements get deleted. So the regulation of this gene will be disrupted. So okay, there is also different other type of this disruption of gene functions. So the first high throughput study, actually the result, there are 2004 and 2005. There are two major groups using high throughput arrays to discover that among normal populations, there is a good number of polymorphism of copy number variations. So this is actually a publication by a group in Computational laboratory by Michael Wigler's group. They developed a ROMA system. Basically, this is short for representative oligonucleotide microarray. To develop that array, you need a lot of bioinformatics work actually to pick the probe sets to represent the genome. And from that study, they actually discovered the number about, they used 20 individuals and discovered about 220 regions. All these regions gain or loss, 220 regions, and roughly about 10 variations per individual. So these are actually occurred in the normal population. So this is important because for this study, when you look at copy number status, copy number change, you need to filter the normal polymorphisms. And oftentimes you need the germline normal control to do the same copy number study, but if not, you may use a pool of all the known copy polymorphisms, copy number variants, copy number polymorphisms. And then actually at that time, I think the array, the sensitivity is not very good. So you see a small set of variants discovered. And Affymetrix developed a SNP array that can do both genotyping, and also you can deduce copy number information from the from this readout. And they provide much more high sensitivities. So this study published in Nature in 2006. We actually look at 270 normal samples, and it covers I think 4 major populations. And they also I think use some HapMap samples for this study. And for this study, they discovered actually more copy number polymorphism regions, about 1,500. Among these 220 patients, normal people, not the patients. So basically, the results show that actually from this analysis, they discovered that the copy number variant regions covers more than 190 megabits long across the whole genome. And so I checked the copy number variants database. The latest version, it shows that now there are more of this copy number polymorphism among normal population. It covers almost 30% of the human genome, and covers about less than 1 billion base pair. So in total base pair, it's actually more than single nucleotide polymorphisms. So this kind of highlights the importance of copy number variations on the disease process or for genetic studies. The thing I want to mention is that all this, the public collections are based on different platforms. Those platforms has different sensitivities, and also their algorithm to call the copy number variants will actually, be different from group to group, and there's a lot of variation there. So the total number here doesn't mean, you will see an increasing number of, genomic regions that are subject to copy number polymorphism. I think in reality that probably should not happen. Okay. So now I think there is a need to analyze large cohort, large data set of copy number variations from public database, from public data set, because usually you have a small cohort of samples that you will have copy number data. If you want to, gain more power, get more statistical power, you want to, put lots of data together. So for that, you need to understand for each platform how they process their data. So I think understanding of the common procedure to process copy number data is essential. So usually the analysis procedure should cover 3 main steps. The first one is the normalization. And there are global normalization, local intensity-based Lewis normalization, invariant set normalization, and also a quantile normalization. And there's a program, DCHIP, that developed by the Harvard group. They use this invariant set normalization very often. And I will show a result that use this program, they discover some really interesting results. After the normalization is done, you need to do smoothing, basically to reduce the small fine substructure of the data, or to reduce the sharp change of the signals. So smoothing is a necessary step to process the normalized signal, and for the next calling step. So for smoothing, there are also different algorithms that you can pick, and the simplest is the moving window averages, the moving window approaches. So you can pick a window size, 1K or 2K, and summarize the SNPs or signals within that window, and for each probe or for each data point, to replace that with its the average of its neighboring data points. And also there are other regression-based methods that you can fit to at least a square regression to reduce the residuals actually to smooth the signal. And also compared to quantile normalization, you have also quantile smoothing. This is kind of aggressive smoothing method. For smoothing, you have the risk to losing signals. Depends on how loose you will set the standard, the threshold. Another one is the wavelet smoothing. It's based on a set of some sort of functions to transform your data, and compared to Fourier transformation, you can actually deal with sharp changes or discontinuities of the signals. Once you're done with the smoothing, you get the signals up and down, and also capture the trend of the change. Then you can do the variant call to call the copy status. So there are 2 major algorithm. There is one called change point method, and also people probably are very familiar with hidden Markov models. So change point method, I think is based on results in 1970, 1980s. Mathematician developed this change point test. So you can basically to see whether the signal changing from one state to the other, whether the change is significant or not. Then you can call that point a change point. You can merge the change point together to get a region of copy gain or copy loss region. There are new technologies like genome sequencing. Using the sequencing data, you can also deduce copy number status, but it's totally different procedure. So the information you need from sequencing is basically the for pair end sequence, you need the pair information of the pair arrays, and also for single end arrays, you need the depths of the arrays. So merging the signals of pair ends with coverage, you can get exact location of the deletion or amplification regions. So there's many genes that were discovered or associated with copy number variation for cancer or different types of cancers. The number is I think it's keep growing if you have if you actually analyze more copy number data set, and also have a bigger data set that will help you to discover cancer-related genes more significantly. So this is a study I mentioned that they used the D-chip method algorithm to study leukemia patients, normal versus tumor, and they use SMP 5.0 chip, actually achieve a very high sensitivity. So for this study, they actually among about 240 pairs, about actually about 30% actually had this gene deletions. So this is actually paired normal versus tumor, all the paired, and also from different subtypes. So these genes clearly compared to normal, you see a clear deletion signals. The nice part of this actually result is that not only you can see the deletion of the whole gene, like for this one, this is a deletion of the whole gene across the whole region. So this gene, this PAX5 gene is about 200K long, and it has a very long introns. So the exons is kind of relatively smaller. And from this information, actually, you can deduce where the intron get deleted. So you can get specifically, which exon get deleted. So you can actually get very detailed information for each individual patient, how their, the transcript of the gene will be like, the PAX5. It's a very important transcript factor. This study is a similar kind of study, similar algorithm setup. So we look at, this is actually a work with Dr. Bill Carroll here in Cancer Center. We look at 20 pairs of patients. They are all patients. One is diagnosed with disease. The other is after some treatment, the patient get profiled at relapse. So using the SMP arrays, we can actually deduce the copy gain or loss for each paired patient. So basically, we can then compare the copy number status between diagnosis versus relapse to actually see where we see gains, copy number change in relapse, some sort of copy gain we see in relapse, but we didn't see diagnosis, or some copy loss in the relapse we didn't see in diagnosis. And also we look at cross sample pairs, the frequency, the recurrent ratio of all those copy number status. So this is a summary of the recurrent ratios for copy gain and for copy loss in different disease in different disease states. This is for diagnosis and for relapse. So from this, actually, you can pick the candidate genes based on their recurrence ratios, and also through comparison of different, the cancer stage, you can actually pick the genes that actually give you some information of how this, the cancer progressed, how they evolved. So this is what I said. When you compare the two relapse and diagnosis, you will see which clones, which copy gain was totally originated from the original clone, and also which clones are actually from the diagnosis clones. So from this kind of analysis, you will have some understanding of how this disease progressed. And this is used copy number data. So recent sequencing data also gives you the information of clonality. If you have enough deep coverage, you can analyze the mutations to deduce, to have actually similar information of how this disease progressed. So similar thing here, you can actually then, if you have copy number data and also sequencing data, you can compare the results for this kind of evolution analysis. Sometimes you have only sequencing data, but you can always get a public data set to compare for your own purpose. So this is actually a result. When you look at individual genes, there are individual genes deletions that common across many patients. So that actually are those genes you see in this figure that they have a higher frequency. So those genes, deserve to be further studied. So basically, this will give you a good, candidate genes for further, functional study to get the understand the underlying pathways or the mechanism of the disease progress. Also use copy number information like gene expression data. You can cluster patients based on their copy number status. So in this result, you can actually see we group the patient into 3 groups based on their copy number change. And also, you can see that it's associated with some clinical outcomes. In this case, it's the time to relapse, from diagnosis to relapse. So there is some certain relationship, correlation there that are related to the copy number status. So each subpopulation or subgroup patients, they share some copy number variants. This is another gene and discovered from this copy number analysis. It's deleted in 3 relapse. So this gene is associated with drug resistance. So these are all the experiment data from the collaboration from Dr. Bill Carroll's lab. Okay, so basically the first part is about copy number analysis, and also now I will talk about some research project using sequencing technologies. So from the same cohort of patients, this is still again collaboration work with Dr. Bill Carroll and Cancer Center. From the same cohort of patients, we selected 10 pairs, 10 patients, basically with diagnosis and relapse samples to sequence their whole transcriptome in order to identify the underlying driver mutations that may explain the drug resistance. This is our original design of the strategy to deal with the RNA-seq data. So we started this project actually 3 years ago. So it's very early at that time. There's not many RNA-seq data was done at that time. So we the idea to use transcriptional sequencing is that we expect to discover not only variants but also differential gene expression, non-coding transcripts, fusion genes, and also for those sequence reads that was not aligned, we were thinking to reassemble the reads and to discover new transcripts. So basically, this is the rough pipeline of our plan to analyze the data. For the variant, actually we tried all this stuff, and there are many results for each purpose, but most of them end up it's not working. Many of them actually are just false positives. The most successful thing is with the variant detection. So for the variant detection, we tried several variant calling algorithms. We use SAM tools, CNV call, GATK. There are many public tools out there. So we tried almost all of them at that time. And it ends up that the GATK actually is a very efficient tool set to help us to do the variant call. So this is basically the procedure of the GATK procedure. If you want to discuss details, we can do this offline. And there's an interesting discussion at that time that posed some sort of doubts on the value of RNA-seq to discover mutations. So this is the first paper published by a group in Science. They compared RNA and DNA sequences from same samples. They actually sequenced up, I think, 20 samples. So they sequenced both their transcriptome and also the exome sequencing. And what they discovered is very surprising, astonishing. They claimed that there's widespread RNA and DNA sequence differences in the human transcriptome. So if this is true, all the variants you discovered, is probably, it may be because of this RNA editing mechanism. And this actually caused hot debates in 2009, 2010, 2011. Around that time, in the major genome biology meetings, people will talk and discuss, and actually there's lots of doubts on their results. So very quickly, there's another paper reanalyzed their data, and they said that actually there are only very few RNA sequence differences in the human transcriptome. So the reason why these two get very different results is because the analysis in this first group is not actually optimal. When they do alignments, so in doing sequence alignment, there are many trivial steps. If you don't pay attention to, will give you lots of false calls. In this situation, the RNA editing sites mainly comes from the paralog gene alignments. So for each gene, you have paralog genes in different locations on the chromosome. When your alignment algorithm you utilize different seed lengths or allowance of a mismatch, it will actually produce lots of that missed calls. So fortunately, actually before they settled this, we already figured out a set of filters on our data set to improve the true discovery rate and reduce the false positive. We already actually we get our the best candidates variants. So later I actually figure out that there was already a study. They compared very quantitatively different quality filters that will affect the variant call. So this study used monozygotes, identical twins, their whole genome, and compare the variant calls from these two identical genomes. So basically, if the algorithm is right, you will get a set of shared variants between these two genomes, and you will have minimum set of discrepancies. So basically, the target function is to maximize the shared calls between this identical genome and minimize the differences between these two variant sets. And basically, these are the major filters that people used. When you look at the literature, you will see people arbitrarily pick one or two, three or four, at most four of these filters for their variant call. Okay, basically I want to also point out that this is for genome variant call, and for transcriptome, for exon variant call, it will be different. Like for this clustered SNVs, so you can look at how the SNVs clusters. You can have an average across the genome, average occurrence of SNVs across the genome. You have a number, and then you look at the locally in the certain range of the genome, how many variants you will discover. So if you see some tendencies of a SNV cluster, those are obviously dubious regions that will give you a wrong call. And so these filters are basically the coverage, the variant score. This is equal to the quality score, the mapping score, clustered SNVs across the genome, near a indel, whether these mutations occurs close to a indel size. And also, you should look at whether those SNVs fall into microsatellites, simple tandem repeats regions, segmented duplication regions, homopolymer runs. So they probably all this kind of all these single nucleotides repeats. And the other filter is consensus mapping, consensus call. Basically, it advertise for that you try your variant call with multiple methods and then see how consensus the results are when you compare different methods. If you have this variant call from different methods, though you have higher confidence that is the right variant call. So there, this study actually look at all the combinations of different filters. So these are 10 filters. So you can have all the different combinations. You have, each single filter. You have the filter or combination of 2, combination of 3, combination of 4, and combination of 2 combination of 10. And use all these different type of combinations. You will check, the discrepancies and the overlap of the calls on these two identical genomes. So basically, you will see the trend that a certain combination gives you basically less discordant calls and more concordant calls. So this is probably the best combination of filters for the SNV call on genomes, but not for our transcriptome. For transcriptome, our experience is for this kind of filter, you need coverage, you need a quality score, mapping score, you need directional information, and for actually this is equal to the allele imbalance information here. So if you have a heterozygous allele, theoretically you should have 50-50, but if you have 20-80 or 30-70, it's kind of dubious sites. So that's the filter for bidirectional information. So there's actually quantitative analysis to determine what's the optimal value for each filter. So for the coverage, you can see the plot that at the depth of 20, you will have relatively more concordant cause and less discordant cause. So basically, you can and also you can check the error rate for at different depths compared to the true variant rate. And this is for the variant score. This is for the distance to the indels. So basically, the idea distance that you don't want to see as SNV occurs closer to an indel within 5 base pairs. This is actually the final pipeline that we used for this relapse sequence project. So this is the filters that actually filter our candidate variants into the final very small number of variant calls. The previous filters are mainly quality filters based on the sequence quality, the genome sequence features, all that kind of stuff. So you also need functional filters to filter out less important variants and keep all the functional relevant, functional important sites. So these are the few filters that I may recommend people to use, when they do their variant calls. This is a final list that we discovered from this study. This is published in Nature Genetics. And also I mentioned the tools we used for the functional prediction, the polyphen, and the SIFT. They are very good tools. This is one single gene in that actually the most important discovery that we see multiple hits on this gene, and also we see that this kind of recurred in about 20% of the patients. Okay, so now I will switch to the methylation data analysis later. From the same data set, we have this is actually a bigger cohort, but overlaps with our previous sequencing and the copy number profiled patients. So we have also patients that profiled by the methylation arrays. So for the methylation study, basically the platform, the Illumina Infinium, gives us the measure of their CPG island, the methylation status of the CPG island in the promoter regions. So basically we quantified the change using these delta beta values, using the beta values to actually quantify the status of each gene based on their CPG island methylation status, quantify them as unmethylated, hemimethylated, and methylated. So when we compare two states, we can actually, for each gene, give them a status, a transition status, whether that gene changed from unmethylated to methylated, or or vice versa, or different combinations. So basically, we use this transition database matrix to quantify all the genes and categorize them, based on their the methylation change. And also similarly with gene expression data, you can cluster your patient with methylation data. Actually, a very nice cluster if you pick the right gene sets. So now, actually, I described very briefly. I hope you all understand, the process for each different, analysis. So we have, gene expression data, copy number variations, methylation status change, and also sequence variants. For some patients, we have measures of all these from all these platforms. So basically, how to integrate this this data together to make some meaningful hypothesis, or integrate this data together to find some target pathways. You know, some sort of driver events, driver mutations, driver variants. There are many discussions on how to integrate all these different data sets. So my actually method to deal with that is to integrate this data through function analysis. So we know the gene expression can be regulated by transcription factor transcription regulation through the transcription factor binding, through the promoter methylation, through the microRNA binding, and also some splicing regulation. So it's coupled with expression. And also for copy number data, through the copy number analysis, you will have idea of gene fusion, exon deletion, promoter deletion, 3' UTR disruption. So all this information you can link to the expression, to the individual genes through the analysis of their functional elements, through their integrated internal regulation, internal relationship. And also similar thing for methylation. You will, if you have this methylation status change, you will check their promoter to see whether there are copy number change on the promoter, if there are mutations on the promoter. And the same thing for transcription binding sites, because this is all about the promoter. So methylation is the change of the CPG island in the promoter region, and also the TF binding sites also occurred there. And also similar things for sequence mutations. So sequence mutation will affect promoters, microRNA binding sites, splicing signals, exonic splicing signals. So all these discrete data sets, they have their internal relationships based on the function study. This figure is my favorite figure that shows the complexity and the integration of the message process, of the message flow from DNA to the final product, the protein. This is actually from the review paper by Tom Miniatis. So basically, we can see from here, all these procedures are coupled. The splicing, 3' polyadenylation, and mRNA export. So the point I want to show from this figure is that the functional integration actually was built internally by itself, by the organism itself. So all this procedure is not separate. So then the integration analysis of a data set makes a lot of sense when you think about the integration process of the message processing. In terms of a detailed operational manageable thing is that for each set you can do canonical correlations. Let's say for expression and for copy number, you will see genes that through this canonical correlation analysis to see whether its expression jives with their copy number change. And also you can do the promoter analysis on the clustered genes and to see how this the mutation effect on the promoter binding sites will jive with expression. And for the promoter sequence analysis, basically, when you have the binding sites discovered on the promoter regions, if you have a mutation occurred on those regions, although it's not affecting the function of the gene, the regulation of the gene was disrupted. Let's say for this gene mutated to whatever C or T, this binding probably will not occur, or maybe at the low efficiency of this TF binding. So basically, the wire will change. I think we remember the last talk from Dr. Jinhua from Stony Brook about quantifying the Wellington landscape. So the foundation for that is have a wiring of the transcription with genes. So basically for cancer genome, it probably will not apply because all this mutation occurred in the promoter regions, in the regulatory regions, will affect actually the transcription binding. So basically the matching of TF with the genes will not apply. So there is no stable regulation networks. So probably you cannot do that kind of quantitative analysis. Also, you can analyze exon splicing signals. So using the tools we developed. So nowadays, when people filter their variants, they will say, okay, whether these variants affect the coding region. So most of the time, if the mutation occurs, it's not changing the amino acids. Sometimes it will affect the splicing signals. It will cause a missing or a gain of exons. Here is an example actually to show the results that are analyzed with the splicing elements analysis tool. So for exon splicing, basically you have signals. This is the traditional GTAG signals and the splicing junctions. You have the branch sites somewhere. And also you have all these intronic enhancers, silencers, and also you have exonic enhancer and silencers. So those sequence elements usually 6 to 9 base pair long. They will help to recruit splicing factors to bind all these proteins, to bind to these exons and to enhance the splicing machinery to include this exon into the final transcript. So this study, they discovered 2 mutations, actually side by sides. And one mutation is a non-synonymous mutation, doesn't change the amino acids. And the other actually changes the amino acids. And when you analyze the splicing signal of these mutations, you will see the change. This is the wild type signals. For the wild type sequence, actually all these 2 splice site signal is not changed. It's above threshold, so it's functional. But if you have mutations, so for this splice site, the splicing signal is actually decreased below the threshold. So basically this is disrupted. The motif is not functional, though probably the binding of the splicing factors is not happening. And for the other one, for this one, it's the same thing, that actually the splice signal is reduced. So this analysis basically, will actually put all these 2 mutations as in the top candidate list, rather than throw them away because of they're not changing the protein coding regions. This is basically the integration in terms of the function integration in my term of a different data set. So there is another way to integrate the data set is to just ignore everything about the function. Just look at this as matrix, as data set. So for that, you can put all this data set from different platforms to improve the cluster analysis of patients. So for this, actually, you can, so this is, so for each different data set, let's say for expression data, when you do the PCA analysis on a simulated data set, let's say you have three groups for the data set. For the PCA analysis on only expression, you will see maybe 2 clusters, the black spot and maybe a cluster, but it's not that clear. And also, you can do the same thing on copy number data. So probably, you don't see maybe 2 clusters, and these 2 actually sort of mixed together. And same thing for the methylation data, it's a little bit better. But when you combine all these different together and using a certain cluster method, you will achieve a better clustering. Basically, this will help you to discover a subgroup of patients that actually are different from the other patients. Give you a more fine structure of the patients. So this probably will be useful for pick a subtype of patients, that may respond, let's say, the response to different drugs. This is a recent study on Nature on cancer, what they call the cancer endometriocarcinomas. I'm not familiar with that cancer, but anyway, the idea is very nice here, that you put all the data, you tabulate them, you have the copy number change, you have the certain gene mutations, and so copy number here, and some mutation profiles, methylations. So okay, this is their icons. So basically, the results from this analysis is that you can combine the different data sets, achieve a very nice subclustering of patients using limited number of variables. For this one, for this I think 400 patients, they actually used only 1, 2, 3 variables. They can partition the patients perfectly. When you look at their expression, they can partition the patients perfectly into 4 categories. And the partition jives with the expression profile. So remember, there's lots of discussions saying that the expression data-based classifier or subtype kind of to predict subtypes using expression, you have lots of multiplicity. So basically, this is maybe 60 genes. So you can use this 60 genes profile to subtype the patients, but also you can pick any certain number of combinations from this list of genes to have similar predictive results. So basically, that tells that you are not using the driver things to predict the subgroups. But if you combine the different type of data, you only need 4 things. Here is you need a copy status basically, and another one is the MSI. This is the microsatellite instability. Basically, the sequence related. Microsatellite will be copy number or instability, and also you have another. This blue I think is methylation-based information. So now the idea is that if you combine the data, it's more likely, if you do the clustering, it's more likely you find the driver events to subtype the patient. This will actually, it shows this expression profile very closely reflect the protein expression, the protein information. So basically, this is another way to integrate data, to use put the data together to find the driver events, the classifiers, the efficient classifiers. Okay, with all this is all I have to say today, and thank CHIBI and the Cancer Institutes, all the colleagues, collaborators for their excellent support. Thank you very much. I hope you get some information back and deserve this hour. If not, we can discuss, and also I think this is a good opportunity that maybe we have some future collaborations or discussions based on these ideas.
What if controversy result from 2 different data sets? How do you integrate them? It happens quite a lot.
What do you mean controversy?
Says mutation or variant data gives you one type of pathways, and the expression data tells you different type of pathways.
Yeah, right. I mean, this is very often you will see the situations that your candidate genes, top genes, the mutation hit different pathways. So for integration analysis, actually, for individual pathways, the effect of a mutation or methylation, they may hit different components in that pathway. So I think it's very legitimate that different cells hit different genes. But when you look at maybe a bigger pathway, you will sort of integrate them into that. So basically, say, okay, they actually different mechanism targeting the components of the same pathways. Yeah, does that answer your question? Or then this is also this idea to do canonical correlation to see whether expression jumps with copy number change. So if they do, it's very likely those are the driver events. So you have the deletion of the genes, you have less dosage of this gene in the DNA sequence, and then you have less expression. But if not, let's say you have high copy number change, low expression. So how do you explain that? So let's say you have copy loss. Probably it's maybe copy loss of one allele, but the other allele get much overexpressed. So that you can sort of deduce maybe something very biologically very interesting happening. All right. Okay, thank you.